Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com, and this week I'm delighted to introduce our special guest on the show today. We have with us Dr. Sam Lee, who is the Co-Chief Executive Officer and President of Cocrystal Pharma. Cocrystal Pharma is a biotech company developing novel antiviral treatments for influenza, norovirus, and coronaviruses, including COVID-19, among others. The company is employing a proprietary structure-based drug discovery strategy to target inhibitors of viral replication. Dr. Sam Lee has over 25 years of anti-infective drug discovery research experience. Prior to Cocrystal, he managed anti-infective drug discovery projects for 8 years at Icos Corporation. At Icos, Dr. Lee played a key role in developing PI3 kinase delta inhibitors, which led to an FDA-approved product. He received his PhD in biological sciences from the University of Notre Dame and completed postdoctoral training in viral biochemistry with Dr. I.R. Lehman at Stanford University. While at Stanford, Dr. Lee founded and was CEO of Viral Assays in Cupertino, California. To learn more about Cocrystal Pharma and its innovative antiviral therapeutics, let's turn to Dr. Lee. Thank you so much for being on the show today.
Thank you. Happy to be here.
So I'd like to start off, of course, by asking more about Cocrystal Pharma, what the company's missions and goals are, and perhaps also what led you to Cocrystal.
Absolutely. Our mission is to discover and develop broad-spectrum antiviral drugs for patients with acute and chronic viral infections. And I think our corporate goal is to, develop and commercialize broad-spectrum antiviral therapeutics. And I think that you asked me about how I got into Cocrystal.
Yeah, I mean, you've been in obviously, you've had a very prolific career in the industry in biotech. So, what led you to co-found Cocrystal Pharma?
So I have to tell you about my training background. When I was doing postdoc at Stanford, I was studying herpes virus replication enzymes with the Dr. Bob Lehman. And I was very fortunate. I had a wonderful mentors that includes Bob Lehman, as well as the doctors Arthur Kornberg and Paul Berg and Dr. Roger Kornberg. You can see the two names, Kornbergs. Arthur Kornberg received a Nobel Prize in 1959, and Roger Kornberg, his son, also received, Nobel Prize. So during my postdoctoral training, I was really interested in focusing on the replication enzyme, which is ultimately very, very attractive drug target, and I was also interested in using, our knowledge, biochemistry, as well as structural biology, how we can develop broad-spectrum antiviral drugs. That's the whole business was started.
That's wonderful to hear, and I think it really underscores the importance of having great mentors, like you said. So that's really great to learn about your journey and how you co-founded Cocrystal. So I'd like to learn more about Cocrystal's technology and its anti-infective drug discovery research, which is leading to therapeutics for various indications. So what can you tell us about the technology and what makes it so novel?
Yes. One of our co-founder, Dr. Kornberg, Roger Kornberg, he worked on structural biology of the transcription machinery, and he did a pioneer work on structural biology of these enzymes, how actually the works in terms of transcription and um, replication, process. Um, During the early stage of Cocrystal, when we developed the business plan, we had two fundamental ideas. One is there are always highly conserved regions of essential enzymes. And then we want to use this information, we call it X, based on X-ray crystallography data. We can see the drug binding sites at atomic level. And now, these days people call AI, technology, but this has been, many years ago, well established. So we're looking at these highly conserved regions of each virus. Um, that once we focus on those targets and apply chemistry, it's a traditional chemistry combined with a computational approach, we can actually predict the broad spectrum binding mode. And based on the computational prediction, we actually conduct the chemistry. So this has been a very successful approach that we this is a foundation for developing antiviral compounds. And I want to just point it out, what are the key differentiating points our technology versus conventional antiviral research work. I think the antiviral space is that you really have to think about three considerations. One is the safety of compound, because this is, we're not dealing with cancer patients. This is a infection that rapidly, develop the disease. So we want to quickly cure, this disease. If it is a chronic infection, we also have to cure this, viral infection without having any side effects. So safety profile is really important. Second important aspect is the drug resistance. Even if you develop the really novel antiviral compounds, that either during treatment or after the treatment, if you develop the brand new resistant viruses that actually transmit to other patients, that's not a really good thing. So when we, as a drug developer, we always have to think about how can we minimize this antiviral drug resistance. We've seen COVID cases that virus constantly mutated. Vaccine number one, the virus everybody immunized, but a few months later, another new virus mutate, and they can escape those vaccine the efficacy. Even small molecule antivirals, same story that we've seen. Tamiflu, for example, that's the first approved influenza drug, and its drug resistance is well characterized based on some other papers. About 10% of circulated influenza viruses are drug resistant. And same similar situation, Zofluza, which known as another name, baloxavir. This is also people detect the resistance during even clinical trials. So this is a really challenging issue as a drug discovery um, team, and we have to deal with the drug resistance. So finally, when we have the safety and resistance, the broad spectrum activity is really crucial because you want to target broad as you can. So one drug could cover all of the existing, viral strains, and if ideally we can forecast that other newly emerging or some resistance, viruses, we can treat those patients with the newly emerging viruses with our compounds. And I think the co-crystal technology allow us to carefully design the molecule they're using this X-ray, high-resolution X-ray data. So starting looking at the potential drug binding pocket. So before even we develop the drug discovery process, we could perhaps predict the broad spectrum binding pockets, and as well as potential resistance viruses. And I think that's a really important aspects of our technology.
And so, what therapeutic assets do you have or have you developed using this proprietary technology?
Sure. Our first molecule was the hepatitis C drugs. That's we complete the phase 2A. Now we have multiple new programs. Second one is, excuse me, influenza virus. This particular compounds targeting highly conserved region of the replication protein we call PB2 protein. This compounds is for treatment of seasonal and pandemic influenza A. And we have this. We developed the oral inhibitor. We're currently conducting phase 2A study. And the next compound is the pan-viral protease inhibitor. We're currently conducting phase 1 study. This is for coronavirus as well as norovirus. And then the preclinical stage compounds that we have, influenza, replication inhibitors. It has a super broad spectrum activity. That's still preclinical stage. Now, one other interesting program we're really excited about is the oral PB2 inhibitors that we're currently conducting phase 2A study, but we develop inhaled compound. So now this is could be potentially breakthrough approach that this compound we can actually use for therapeutic as well as potentially prophylaxis. So if you think about the significance of this prophylactic approach, during COVID time, if you have a pandemic infection, if you have family members got sick or hospital situation, any places that you are exposed to infected people, perhaps this drug candidates by inhaling, it would be prophylactic use for the infected dealing with the infected patients. So we are hoping to advance this program to phase 1 study this year. So currently we're conducting GLP tox study.
Wow, that's really exciting. And to have like an inhaled version, we are hearing more and more about different variations of like vaccines as well, oral vaccines and different drug formulations. So that is indeed exciting. And what are the current unmet needs and pressing needs with respect to infections from influenza, norovirus, and of course SARS-CoV-2 that leads to COVID-19 and other coronaviruses? I think we kind of know what these viruses do, but how does it translate into what's happening on the ground, and what's the current landscape with these infections and these viruses?
Yes. So even though we just deal with the COVID pandemic outbreaks, it's really terrible. But I'm really proud the pharmaceutical business, U.S.-based pharmaceutical business. We did a pioneer work on vaccine development as well as antiviral development. So if you think about the fundamental problem is the virus, these RNA viruses or DNA viruses, they constantly mutate. So if you think about, COVID case, this is not the first time, pandemic outbreak. 2003, in China, there was a pandemic outbreak. So now we're going to call it, SARS-CoV-1, SARS coronavirus 1. So that was a pretty significant, pandemic outbreak. But, you know, we didn't have a medication. We didn't develop the vaccine. We just went on and, we just forgot about it. And now, SARS coronavirus 1 that mutated, now it's a SARS-CoV-2. When you deal with this mutated, newly emerging, viruses becoming pandemic, we're really, we don't have a lot of options. If you think about very beginning of the COVID pandemic, we're trying to use anything we could use, repurposed drug, anything that we can use. But if you think about it, this is gonna come back again. Influenza virus, we know famous story, Spanish flu, millions of people died. But if you think about it, how many drugs we developed since Spanish flu? Well, there are two approved drugs that are widely treating influenza patients. Tamiflu is an old drug approved back in 1999. And then second, newly developed, newly approved drug, Influenza. Again, both the drugs have really, remarkable, pharmacological properties, but resistance issues is not going away. So if you think about now with, news, you've seen news articles, avian flu in Texas, farmers already confirmed they have this avian flu. So if you think about the virus that resistance to existing drugs, right? In brand new virus, and then came from bird and infected human. This is again well-known, process. So if that happens, we really don't have a lot of options. So I think we have to constantly develop new medicine, new vaccines, and it's more importantly, we have to develop, really precision, medicine. I think a Cocrystal technology use our X-ray data to design the molecule computationally and as well as experimentally design the molecule that will work targeting the most conserved region and functionally absolutely require this for viral, replication. If you block that, the virus cannot, mutate those regions because it's essentially required. So our approach has been really looking at the, we call drug binding pocket that, the potential broad spectrum compounds binds that certain area that we computationally again predict can virus mutate these residues. So we use those computational data trying to forecast that the virus cannot or could mutate these regions, and that's how we develop the compounds. Not only broad spectrum. Now we're talking about pan-viral. So coronavirus and norovirus is completely different RNA viruses. However, compounds that we're developing because we focus on most essential and conserved region of the, regardless which virus we're talking about here, that area. So it's actually translate. It's a really, big idea, if you ask me, because that actually we start with the simple broad spectrum, best in class, molecules. Now we extend our capabilities to pan-viral compounds. So these compounds, I think, is brings a huge benefit for patients because that we cannot really focus, brand new viruses because the virus has their own way to mutate. So only way we could do is that we could have a super broad spectrum pan-viral medicine that will work in case that some brand new virus will pop up. So that's our brand new strategy, and I think that we're so excited about the first in class antiviral compounds against the noro and coronavirus infection. Norovirus, there is no vaccine, no antiviral drugs at this point.
That's truly exciting, and actually, it's a great kind of stepping back, big picture strategy where you're targeting a highly conserved, regions and parts of the replication machinery of viruses that are common to all viruses. So you have this broad spectrum therapeutics that can work against multiple viruses. So that's very, very exciting. And so what can we expect from Cocrystal in the coming weeks, coming months, and this year? What are some of the key milestones that you're looking forward to?
Yes, short-term milestones this year, we will have top-line data from influenza PB2 inhibitor. This is a currently phase 2A study. This particular study is a human challenges study, so we recruit healthy volunteers and infected with the influenza virus, and we treat these infected, patients with our drug. So top-line data will be available this year, and the second top-line data will be also available from noro coronavirus trial, phase 1 data. So far, both the studies are going really well, and then the third exciting news would be inhaled influenza compound that we're going to initiate the phase 1 study.
Wonderful. Dr. Lee, any other closing remarks or to round out the show?
Well, thank you for having me. I want to share, our exciting news, but at the same time, we just have to, think about future pandemic, preparation. Mm-hmm. How are we going to deal with this, brand new pandemic infection? I think the awareness we need to develop, novel vaccines, novel drugs. This is going to be an ongoing effort, but I think that people, people should not forget, the COVID is not completely done. Virus will come back, unfortunately.
Exactly. And Dr. Lee, thank you so much for the work that you're doing in the field to help lead the initiatives to combat these viruses and infections that, like you said, like COVID, won't be going anywhere anytime soon. And we'll probably be seeing more of them. So thank you for all of the work that you do. And we look forward to learning more about and to seeing those milestones that you mentioned that will be coming up this year in terms of the top line data that will be coming from Cocrystal. So once again, Dr. Lee, thank you so much for being on the show today. I greatly appreciate your time and your insights.
Thank you.
All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we'll see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.